right, we're gonna hide the floating panels. We'll see if Joey's advice works. And so today's lecture is the second part of DNA. We're gonna really be focusing on the RNA. And so this is, we talked about DNA in the last slideshow. So DNA transcribes, or it's called transcription, to become a sequence of RNA. So since they are both nucleic acids, it is called transcription because you're just transcribing within the same language. And then the RNA is the working drawings to make of the code for the protein. And that is called translation because it's actually a totally different language. It'd be like going from English to Sanskrit. So we're going from nucleotides to amino acids. And this whole thing was called by Watson and Crick in their famous paper in the 50s, the Central Dogma, which some people have said is quite arrogant because dogma means creed, or but it has become, you can buy t-shirts, you can buy all sorts of stuff. And I do recognize that probably half of you know this and could teach it, but maybe listen along and you might learn a little bit different perspective. So again, DNA to RNA to protein. And this is how it is traditionally taught and still taught, as the t-shirt shows. One arrow, one direction. Something I will talk about at the end of the show. Is this correct? And actually it is not. The flow is not one directional, and we will get to that. But I'm going to start with where our knowledge base still seems to be, which is DNA transcription becomes RNA. RNA then is translated into protein. And it is absolutely very cool and awesome that we figured this out. And again, DNA's structure was figured out in the 1950s. And so this whole thing is not that old that we've understood it. All right, so this is a picture of transcription. And again, I apologize. Usually I have the board and I can be writing as I'm doing this. But transcription, again, is the DNA. So this is DNA because it's two strands. It's our famous double helix. This is RNA. It's going to be one strand. And so this is, people spend their whole life just trying to figure out one piece here. And so this is this little piece. This is the sigma in there is a factor. So remember we talked about cofactors and coenzymes. There are a lot of them. This is way oversimplified. But the big green triangle is RNA polymerase. So anytime you see an ending ASE, that implies the enzyme. It's the enzyme that makes a polymer, continuing huge sequence, and it's going to be of RNA. DNA is the template. RNA is the product. RNA polymerase is what does it. There's three basic steps. We initiate, so we begin it. We elongate, which is the nucleotides are put together, and then it ends. And why that is important is in you and I, our DNA, again, only 2% is coding for RNA to protein. So we don't want to, remember there were 3 billion bases. It would take you years to even be able to do all 3 billion. And we want to make a new protein in a lot less than 95 years. So we only want to transcribe a small portion, a gene. So again, a gene is a segment of DNA that codes for a specific protein. So that's what's happening here. The initiation is, there's all these factors that are called transcription initiation factors, and they say, hey, let's start here. And then the polymerase comes along, goes, okay, and makes it. And then there's things that are called termination factors that say, okay, let's stop here. So that it only has to make a very small amount of it. So it was a question on your quiz 9. So DNA is huge, 3 billion base pairs in us, and RNA, you know, they're small. So anywhere from 50 to 1,000. Another thing I wanted to point out is this elongation step. What is really cool about it is what happens first, and I mentioned this with DNA polymerase. So DNA polymerase is what made the two strands of DNA. One is you're making a complement to the DNA, and it is that attraction of the base pairs. So the bases, the nucleotide comes in and is attracted, so G goes to C. A would now attract a U, a uracil, because in RNA there's no thymine. And then the polymerase hooks the backbone together. So the backbone is the sugar phosphate, and that is what the polymerase is doing. So the attraction happens first, and then the polymerase comes along. But this is happening like 9 nucleotides per second. If I had you guys try and put strings in the correct sequence on a piece of thread, there's no way you could do 9 in a second. And so this attraction is huge. And then the polymerase comes along and elongates it, which means makes the covalent bond. You always need an enzyme to make the covalent bond of phosphate to sugar. The RNA is growing 5' to 3'. So the polymerase is going along the red strand here. It's only going along one of the strands. And so it's moving along from 3 to 5 because this is growing 5 to 3. There's all these details we could go into, and I'm sure you get to take classes where you go into some details. This is just somebody's image of the polymerase. So it's like this, right? The huge enzyme, there's the active site. The double helix tells you it's a DNA. And then here's the RNA that we're making. And so only part of the DNA gets unwound, only the part that we're working on. And again, you can see this is growing 5' to 3' as the polymerase is moving. And great. And then it knows to end. And here's the next nucleotide gets attracted, and the polymerase is going to make the bond, right? There's all this extra information we're not going to worry about. It's just a big picture. This is a wonderful picture because, again, I'm a biochemist. This is the protein because you see all the alpha helices. It makes a tertiary structure. Question on your exam. All of the protein is important because it determines the overall tertiary structure. See all those alpha helices. And then here we go. This is the DNA because you can see the double helix. All right. So let's talk just briefly about prokaryotes and eukaryotes. And again, some of you I know have had biology and you know all of this, as you will tell me, which is great. And some people have not had biology, and so that's why I have some of these. So prokaryotic cell is a bacteria, and they have no nucleus. And all of their DNA is transcribed. So all the DNA is made into RNA. And you and I, we are eukaryotic cells. So there are all these different kingdoms now, but you and I have a nucleus. Our DNA is in there. And so there's an extra step we have after transcription happens. So the gene gets transcribed. This is happening in the nucleus. Has to be in the nucleus. Why? Because the DNA doesn't leave our nucleus. And this picture also I liked because it points out, right? We have all these extra pieces to us, and every cell is going to be different. There are, again, considered in somebody who thinks about DNA, you're either in the nucleus or you're outside the nucleus. So inside the nucleus is the nucleus, and outside is the cytoplasm. All right. And so there is an extra step, and it is called splicing. And this step only happens in eukaryotic cells, and it happens in the nucleus. So before the RNA can go to the next step, we have to splice it. And there are 3 different types of RNA involved here. So type 1 is called heteronuclear RNA, and that complements the DNA sequence. This is key. It is a complement. So remember, we complement each other. Oh, I like your outfit today. It's very bright and cheery. Thank you. All right, it complements the DNA, and we're going to do practice with this in a little bit. And it is before the splicing happens. So this is the original transcript. This is what you get from transcription. This used to be called the pre-transcript RNA, but they've changed that name. But I mention that because, again, some of you have had biology previously, and so you may have heard that term. And again, I do apologize. It seems now there is so much information. Again, people will spend a lifetime on just one little step here, and then they come up with a new term. But these are terms I'm pretty sure your book used. I think your book may have used pre-transcript. I actually like this term better. It's called heteronuclear, and we'll see why in a moment. There is what's called small nuclear DNA, so snRNA. So the RNA is always capital letters, and the other one or two letter is lowercase letter. So SN stands for small nuclear. And so it is also only found in the nucleus. So these two are only found in the nucleus, and it is small. And it is part of what's known as the spliceosome. My favorite name for them is the SNRPs. So it sounds like Smurfs. And these guys are going to be doing the splicing. And what that means is they're going to remove introns. And we'll see a picture of that in a moment. A spliceosome is made up of both RNA and protein. So it's a complex of both RNA and protein. And so it acts as the scissors. It's going to cut out the introns. And so what are the introns? Well, the messenger RNA is what we're going to end up with. And the messenger RNA is going to be exons only. And the exons are the code for the protein, plus a little bit extra on each end. And we'll maybe talk about that if I remember. That's a picture of the SNRP. So this is RNA. It is not double-stranded. It loops around on itself. And so it can base pair to itself. And then these are different protein complexes. The whole thing comes together and makes this one thing, and it's going to cut, and it's going to remove the introns. So the hnRNA is made up of exons and introns. The scissors, the middle guy here, is going to cut out the introns, and then you're left with the messenger RNA, which is only exons. And there's another slide that I will show this for those of you who've never seen this process. Bacteria don't have the introns, so they don't have to do these steps. So when I was in school, we didn't learn about, we did have this step, but it wasn't in as much detail because a lot of the focus was still on the bacterial part. So that's a picture of it. This is the pre-messenger RNA, which I was calling the hnRNA, and it has exon, intron, exon. This is a very simplistic slide. And then we're going through splicing here. So the intron, just like splicing, Joey had to edit some of my videos, and so he goes in and cuts, and then puts them together. It's the exact same idea. It's going on in your cells, in the nucleus of all your cells. It's so cool. And there's the SNRPs, and they're coming in doing their thing. They're made up again of protein and RNA. The RNA is called small nuclear, and we get the mature messenger RNA. All right, so that's splicing, and it happens in the nucleus. This is cool stuff. You don't have to write this down, but this is something I actually just learned about the week before we started class. And you guys are going to get to hear about this because Lab 9, you're going to get to listen to a talk. There's really no graphics. It's not really a documentary, but it's a phenomenal talk. And I decided to do it at the end because it refers to a lot of things that we've been talking about all term. But and also, if you're trying to look for a topic, this might be too big of a topic though, because it's pretty complex. But what they're showing is this is the gene, and it gets cleaved or spliced in different ways, and it makes a whole bunch of different really important pieces. So what I'd said previously is actually oversimplified. We are actually very complex. So even though only a small part of our DNA codes, the part that codes in us as humans does some really cool things. And so by splicing in different ways, so this gets coded, this gets transcribed, and then your body decides what part it wants. So it then will splice out the part it wants, or it splices, and then that part continues on to the next step, and the other part we're not going to worry about. So some of the things, again, this is extra information. You don't need to know it, but it's really cool. It's something we figured out. So endorphins, remember these were the ones that are our pain response. There's alpha endorphin. Beta endorphin is really cool because it makes a loop. It also can produce the beta lipotropin, which is then a precursor for this one and this other one, which releases fat reserves from the liver. So if you have this one, if you don't splice it a second time, that's what it's going to do, is release fat reserves, which could be a good thing because that means you're going to burn them. But if you're dealing with pain, it's going to get spliced down to this. And ACTH, if you take anatomy, you learn about that. ACTH says, hey, I'm stressed, let's release the cortisol, and cortisol starts making glucose from your amino acids, meaning it starts breaking down your muscles. So whenever you're stressed, you start breaking down muscles to make glucose, and then your body stores it as fat. So really interesting. This one is saying, let's use the fat. This one's saying, I'm stressed. All right. MSH over here is, again, more than you need to know, but I thought this was really cool. Melanocyte stimulating hormone, which is our skin pigment. And really interesting, the same hormone is not only the coloring of your skin, but also determines how hungry you are. So leptin's a really fascinating subject. Students who've taken like nutrition, you learn oodles about it right now because, and again, this is from 10, 11 years ago. So there's actually even more. And then you get CLIP, which is this other hormone. And then you get JP, which is not shown on this one, which is joining peptide. So if you're one of those people who still doesn't have a topic, gosh, look into one of these and say, hey, that's really cool. I want to go into it. But one of the things that's shown here, depending how this is spliced, determines if you're nice and slim, like my puppy, or, oh my goodness, we are dog sitting right now. And Milo, I love Milo. Milo is the friendliest dog. I haven't seen Milo in a year. Milo's diet, Milo is fasting right now in my house. Milo is so sweet. Milo is eating regular dog food apparently, and the people who take care of him, oh, they love him, and they are such beautiful people, and they are thin and slim, and their dog is obese. And yeah, it has to do with how these things get spliced, which means what determines how it gets spliced? That's really the cool question, and that gets into epigenetics. What you eat and your attitude. This is another really cool thing that I found. There's something called microRNA. I'm like, what the heck is that? We don't need to worry about this one, but we're going to worry about the 5 main types of RNA right now. And then, you know what, when you take this class, or maybe you'll be the teacher of this class, there's going to be like 100 different types of RNA suddenly. So these guys are really small. And there's over 600. They keep finding new ones. We keep finding new things because apparently we're changing as a species. And they're really tiny. They have different names. So exogenous are the ones that come from outside. So you are exposed to them, somehow they end up inside of you. There's this one called Peewee RNA. I thought that was cool. So what they're important for is after transcription happens, the RNA gets made, these guys go on and they change something after the transcript. So this is your protein, this is RNA is getting made, hooray. Oh, we're gonna splice parts out. All right, and so what it does is this little, see that little loop there? That's like the Peewee RNA or the siRNA, and it's binding to RNA because it complements it. So A to U, so that's what it's trying to show here. It's making a complement, and it silences the mRNA. So the mRNA is there, it's been made, but it can't do anything. Kind of feels like, sometimes you guys feel like you're silenced. It can silence it in different ways. It could cut the mRNA into 2 pieces. That's one of the things. This is our next step translation, which we'll talk about. It can destabilize it, so it can actually shorten it. It can make it so the ribosome can't do its thing, which is our next step. Kind of crazy. There's all these ways. And I think about 60% of our genes have this post-transcriptional regulation? They don't really know, because this is another hot new field in the past decade. And they also don't know if it's found in other species, how far we would look at that. And then there's another one, and I just like this picture, so I used it, and it's called that RNA can determine our proteins. So it was something a student had said, and she'd come out of her biology class and was all excited about this. So it stands for SHIST, which is X inactive specific transport. So they're calling them RNA genes that they're found. This one specifically is found in, so you and I, we have placenta when we're inside mom, so elephants also. And these are huge transcripts. They're like some of the biggest ones in our body. So 17 kilobases, 17,000. And it gets spliced. It gets that poly A tail that you may have noticed in the last slide. There's like all these things that happen to the RNA. And when you take biology, it really depends who your biology teacher is and what they are really into at the time. Kind of like you with your biochem teacher, right? That would be true for all of your classes, in case you never realized, and everything you do in life. So even, all right, but it doesn't get translated. And then it just coats the chromosome, and it's expressed only by the inactive X chromosome. So we have females have two X chromosomes. So the inactive one gets a coating, and yeah, it's like a sleeve that goes over it. And anyway, then we have these, which are chirps. There's all this stuff. Anyway, let's move on. Translation. The other name for translation is protein synthesis. So the other name for transcription is RNA synthesis. Translation happens not in the nucleus, so in the cytoplasm. And I love this drawing. So this is the DNA gets transcribed into the RNA, gets spliced. That's all happening inside here, the nucleus. And then this RNA leaves the nucleus. So you can see it's leaving by these little tunnels. And it comes out here. And out here in the big world of the cell, there are these awesome complexes called ribosomes, and they're going to perform translation. So that's what I just said. The RNA is translated into protein sequence, and it's done by the ribosomes. And there are, like, I don't know how you can count the ribosomes. It's a question your homework said, and it says how many ribosomes are in a cell, and you can Google whatever the number is. It's a crazy number, and I don't know how you could count it. I actually do know how they counted it. It's just an estimate. So they just looked at one little part of a cell and then one little other part of a cell, and they counted. But nothing is static in a cell. Everything's always moving around. This is more information than you need, but these little canals are called the endoplasmic reticulum. And so some of them have ribosomes. They're just covered with ribosomes. This picture does not do its surface. And then there's also ribosomes all over. You can see a ribosome with a regular light microscope. You don't need a really fancy microscope. They look like little black dots. But they're huge complexes of RNA and protein. And it's a special type of RNA, that little rRNA. And again, some of you have had all of this, and some of you have not. And those of you who have not, when you do this in another class, you'll be the smart one, because you'll be like, oh yeah, I've heard this before. This is kind of cool, extra information, that actually a lot of antibiotics work because they're able to block the ribosome of bacteria. Their ribosomes are slightly different than ours, and so that's how the antibiotic works. But again, 53% of the cells in your body are bacteria. Those antibiotics will affect them too. And so they're finding there's a lot of complications that can come from taking antibiotics more than you really need. Our body's amazing. All right, so these are proteins. That's what it's going to make. It gets folded. This is one of the steps that they don't fully understand, because it just knows how to fold. And good vibrations. Keep good vibrations, good attitude. Smiling at your teacher when you're talking to her. Some of you are really good at that. RNA is involved in translation. So we did this slide with the splicing RNA. That's a picture of a ribosome, as is this. So this piece is the RNA. These would be the nucleotides, the bases sticking off, and then these are amino acids. And right, so this is the ribosome moving along the RNA, and it's churning out amino acid sequence. All right, it's important. This is an enzyme complex because to put the amino acids together requires enzymatic activity because it's a covalent bond. All right, 3 types of RNA involved here to overgeneralize this. mRNA, which is the messenger RNA, so that was the end of splicing, and that is just the exons, just the part that codes, and it's the pattern to make the protein. All right, the t-RNA is transfer RNA, and it is the smallest RNA, except now that they have the peewee and stuff like that. But in our homework, we're not going to worry about those. So transfer RNA gets t-RNA, and it is transports or transfers the amino acids to the ribosome. So somebody has to bring the amino acids to the ribosome. And then the third type is the rRNA that I already mentioned, stands for ribosomal RNA, and it's part of the ribosome complex. The ribosome's huge. Again, you can see it with a regular microscope. And so this picture, this is the DNA, and then these are the 3 types of RNA involved in translation. So that's the messenger RNA, which is just part of the sequence we need. This is the ribosome, has the ribosomal RNA in it, and then there's a little transfer RNA. Should be shown really little. All right, it's out of proportion. It's way out of proportion cartoon. So another image, right? Looks like a jellyfish. That's somebody else showing the ribosome. The ribosome, more information than you need, but if you like to have a lot of information, I could load it on pretty heavy and deep if you want me to. If you never heard, that's what PhD stands for, piled on, heavy and deep. So, but the ribosome has 2 units, and right, my doctorate actually was translational regulation of ornithine decarboxylase, and if you want to read my dissertation, it's right here. Joey's never read it. But at the time, not much was known about translational regulation, so that's why I picked it as my topic, because I figured it would make me sound super smart in front of my committee. It worked. Got a PhD, right? But and since then, a lot more is more and more is being focused on all these, like, new layers are always added, and we keep going. We can go more and more and more into depth of all these things. All right, RNA. There are things called codons and anticodons. And so this is really the question. You don't have to write down this question, but the question becomes, for those of you who ask questions, we have 4 nucleotides, A, C, G, and U. So remember, we're talking about U now because we're talking about RNA. And there's 20 amino acids. There's actually more than that, but we're only going to talk about the 20. So a codon is 3 nucleotides. Nucleotides are talked about as bases on the messenger RNA that codes for one specific amino acid. So AAA codes for a specific thing. Actually, UUU, excuse me, codes for phenylalanine. And so it was Nuremberg made little sequences of RNA. So he made one that was all U. Uracil, UUU, and he fed it into a bacteria, and all he got out was P. And so he's the one who figured out the code, the universal code. The anticodon, need to know what codon, anticodon is transfer RNA. It's on the transfer RNA. It's 3 bases again, and it complements the codon. So if our codon was UUU, the anticodon would be AAA. So it's part of that. Remember, the transfer RNA is like 70 nucleotides long, and so there's just 3, and we'll see a picture, I'm not sure where in the slideshow. So if you take 4 bases and you put them in groups of 3, you have 4 to the power of 3, and you get the magic number 64, which is historically how many tomato plants I would put in my garden, but my garden is overflowing with a plethora of other things right now. so my geekiness, but 64 is a magic number. The thing is there is actually only 20 amino acids, so why would we need 64 codons? There is something called redundancy. In the next slide I will show it, but I wanted to mention for those of you who've never heard this, that number 64 showed up thousands of years ago in one of the most famous books, which is called the I Ching, or if you want, because we're American, we can call it the I Ching, which is the Book of Changes, which there's 64 tangrams, which are these lines, which comes from Taoism. And yeah, there's 64 possible patterns you can make, and it was a way they would predict stuff. So here it is, the universal code from Nuremberg, and it is, there is only one universal code. There are many different ways it is shown out. I like this one because it feels more like a code. This is actually how it was always when I was way back in school, was shown this way. They've dumbed it down, and so they'll put you, but this is going to be the first one. So if we're talking about you, and then the second is you, you're now in this column, and then the third one is you. So you go you. Let's say the code was UAG. You would say, okay, I'm here at U. I go under A as my second position, and then I go to G. You go, oh, stop. What's that mean? Well, we have to stop this at some point. And so if you notice up here, there are 3 stops. Redundancy is if you look here at Lu, if you start with C, and then you go to U, and then whatever the third position is, it's always going to be leucine. Those of you who've done this before, you're like, you've left the room probably. Those of you who have not, we're going to watch a little video where I walk you through it. We'll do some practice. And it is one of the coolest things because we figured out the code in our DNA. And so let's watch. So unfortunately, that disappears.
So we are going to make we're going to make a DNA change into RNA, change into protein. We're going to do it together. And on your study set 10, so the second study set for nucleotides, there is the universal code. And in the slideshow, it will also be up on there. There is one universal code. There are many different charts of how they lay it out. And so I realized that I'm in an interesting situation because you can use any chart you want. But I'm going to use the chart that's here in the homework because to me it makes the most sense. All right, what we're going to do is we're going to start with the DNA strand. And I'm just going to make up the strand. So we'll say gag, cat. Cats gag a lot. And tack. All right, and then we'll say 3'. Now, molecular biologists, biochemists, like to write in 3s, and that's the codon that we're going to get to on here. But before we get there, we have to make the other strand of DNA. So always the complement is opposite. So the 5 becomes 3 and 3 becomes 5. You're always going to label your N. And anywhere you have a G, you would write a C. A's become T's. So my GAG is now CTC. My cat will now be GTA. And my tech will be ATG. All right. It's DNA because we have T's in there. So now we're going to make it into RNA. Oops. I can spell RNA. Capital letters, ribonucleic acid. Now, DNA has 2 strands, and they were classically called the sense and the antisense. But what happened was a lot of people who didn't actually have training in this, they always got it backwards when they taught it, so they changed the name. And then what happened is people still got backwards. And so now it's even more confusing because it seems like every teacher I know here calls it something different. So I'm going with what I'm pretty sure your book calls it, which is just what's ingrained in my head. So in the DNA, one strand is going to be called the info strand, and one strand is going to be called the template. And I will always tell you which one you're starting with. So you're starting with the info or the template. And the key is this. The RNA, the info, it comes out the same as the info strand. So we just go back up there to what I labeled info, and I say, okay, it was 5', and I just write gag, cat, and tack. And you know, it's so awesome, I'm on tape, so I can make mistakes, and I made one on purpose because you should all be going, oh my gosh, she made a mistake. What's the mistake I made? So it does come out the same as the info, except, and those of you who didn't catch the mistake, hopefully you're catching it now. Anywhere there was a T, erase that T and make it a U. So I actually recommend, having done this now for many, many years, that you actually just rewrite the info strand exactly how it was, and then go back and just erase the T's. Now something very interesting happens here. If you remember with our DNA, that word complement. The two strands of DNA are complements. Remember? We are complementing each other. John, your hair is looking extra special today. And Joey, wow. Loving the man bun you got going on, Joey. Joey's desperate to get his hair cut. So, anyway, they complement each other. They're always, that's why your DNA is always so long, because complementing is an act of gratitude, and it actually lengthens our DNA. And I get into that in this slideshow. I may have already. All right, so what that means, the template and the RNA are also complements. So anywhere there was a G, it became a C, the T became an A became a U. So that is actually how your body works. That's how RNA polymerase makes it. It makes it as a complement to the template. It doesn't actually go, oh, there's the DNA info, and makes it the same as that. But you and me, because of how we think, we're not little RNA polymerases. The easiest thing is to go up and say, okay, the DNA info and the RNA are the same, and because they're the same, they complement the template. That is why it's called the info strand, because it gives you the info that is the RNA strand. And that is why the template strand, the word template means it is what the other strands were built off of as a complement. So that's why DNA polymerase and RNA polymerase actually are quite fascinating, because the polymerase doesn't happen first. What happens first is the complement happens. What beautiful eyes you have today. Who says this, Shanti? My dog has heterogeneous eyes, meaning two different colors, and they're just gorgeous. So blue and brown. All right. So the complement comes in, and then the DNA comes along, and the polymerase comes along and ligates the backbone, the phosphate to sugar, phosphate to sugar, phosphate to sugar. So the phosphate and sugar are together, but the next phosphate sugar and the next one. So we're going to do one more step, and we're going to do the polypeptide. And for the polypeptide, we read the RNA. So, and we read always 5' to 3'. That is important. That's the step, the most missed step, is it has to be read 5 to 3. So I actually recommend you put little arrows. Nobody follows my recommendations, but I'm going to recommend it for you, because we're going to do another example in the minute, and the RNA is going to come out going the other way. And so you're going to have to read backwards, which is fascinating. All right, gag is gag in both directions. And so reading this, the first column is, it says the first position is G. So I go down to the little box down here, and this is so much easier when I have that up there. And then I find the A, which is the second position. So I'm in the lowest 4 rows where it says G. And then I go to the third column where I'm now under the A. And the very bottom one, the third position is G. So it says I have glue, which is glutamate, right? And then I find C, so I'm in the second set of boxes, and I go over again to the third column, to the A, and then I come down to U, which is the top. So it says his, like a snake, which is one of the secret sounds. Chinese sounds. Just this. All right, and then U is the top 4 rows. We start there, and we come over to A. Oh, they all have A in the middle. That's because I was trying to spell something. All right, U, and then we come to A, and we come just 2 rows down, and C is tyrosine. But wait, there's more. We have to label our ends. The 5' end of the RNA always corresponds to the N, or the amino terminus, of our tripeptide. These guys are hooked together. You can make a line, you don't have to, and then this is our carboxy terminus. That's my amino terminus, the N, and the C terminus, so gluhistide, the C. It's a tripeptide, it's small, but remember neuropeptides, and so it might be a neuropeptide that's doing magical things for you right now. We're going to do one more step with this, and then we're going to change it around. We're going to do something called a silent mutation. I tell you, I don't think I'm ever going to get over lecturing in front of an empty room. It's very peaceful, but I do miss all of you. All right. Silent mutation is when there is a mutation but no change to the peptide. So no change to the protein or the polypeptide sequence. So if we go up to GAG, if we look, find where GAG was here, there were 2 things that said glue. So as long as I had GA, this G could have been GAG or GAA, it turns out. So if I had mutated that to A, it's still glue, still glutamate. You only have to give me one, but I'm going to give you another example just to make sure, since I'm not with you. And this is beautiful. So this is the code. Humans are really into codes. We're really into numbers and codes, and this is the most magical code of all, and it's in every cell of your body. It's in every cell of your dog's body, or your cat's body, or the nematodes out in the soil that are making it beautiful and making lots of awesome tomatoes. Everybody has the same code. And so the argument goes, is it because we evolved and the code evolved and came along with us? Or is it just one master blueprint? Does it really matter? I don't think it does. Anyway, if we look at CAU, there were 2 things that could have been histidine. So we want to keep it as histidine, but we could have been CAU or CAC. So this could have been a G or a C. I'm sorry, a T or a C up there, and so on. Yeah. Now, let's do a devastating mutation. So that would be a silent mutation. We kept it as that. Let's do a devastating one. Oh, no! Like silent is, well, silent would be like you're in peace. You're meditating. You do your own. There too. Okay, you're peacefully meditating. You have your third eye wide open if you want. The devastating one is not so good. We're going to do it on the last one. Because if you look what was UAC, right under it says stop. So what if I changed it, that C to an A? So what if this became an A up here? So if it had said TAA, this would be UAA, and UAA would be, oh, that gets changed to a stop. There's 3 stops on here. And stops are important because they say to the ribosome, stop, you're done. But in this case, well, we don't actually know if it's devastating. We call it devastating because we no longer have a tripeptide, we only have a dipeptide, but it's possible. Maybe that dipeptide actually does a lot of good, but it's different. So maybe we should call it a different mutation. All right, we're going to keep going. Gotta get my eraser. And, all right, I'm going to erase. You guys can stop and go back and rewind. The magic of the internet. And our original strand is going to be the exact same thing. We're going to do a gag-cat-tack. Still 5 to 3, but I'm going to call this the template strand. And we're going to get a different tripeptide. All right. So, this is still CTC, GTA, ATG, 5'. I actually recommend that you stop me, should have said this before, and you try it yourself. Stop me. Playing freezing statue. This is the info strand. So again, I will identify where you're starting, and it comes out the same. It's a complement. It just puts our perspective. All right, so we make the RNA. I'm in trouble. I keep going in my rant, like I want to run away. The RNA is the same as the info strand, which means the direction is the same. So the tree is there, and we write, like I told you, to write it exactly the same. And some of you are like rolling your eyes saying, oh, I'm so much smarter than that. You would not believe the mistakes I see. And then go back anywhere there was a T, erase your T, and that makes it EU. So our RNA is now different. So our RNA, we actually have to read it in this direction. Now, I have learned, after 25 years of teaching this, I can read that backwards. It's GUA, AUG, CUC. CUC especially is easy to read backwards. What I've learned, less than half of you can do this correctly, that about half of you have to flip it and go, okay, I have to change it so it's GUA, AUG, CUC, and then you can read it in the forward direction. And that's fine. I'm going to do it both ways, and we'll see we get the same answer. As long as you label, the 5 becomes the end, and you would find GUA on here, and G, and then U, and no matter what you get under G and then the column of U, so again, G is the bottom 4 rows, you get valine. And you can write backwards if you want. Wow. Or down here, you would have, this is your N-terminus, and this would be valine. All right. AUG, I know what that is. That's methionine. So AUG, so that would be M. I'm writing backwards. I hope some of you are doing that too. All right, and then methionine. Sorry. Maybe you don't hear the squeak. And then CUC, no matter which direction you read it, it looks like a CUC. So CUC is going to be loop, like skips my loop. That's what I always think of there. And then this is our C-terminus. Right. So valine, methionine, leucine is our tripeptide. And if you look at that last piece, CUC, CU, if I change the C to anything, it's that last position. This is called redundancy. So this could be an A, a G, a U, or a C, and no matter what, we get leucine. This is called redundant. It allows for a mistake to happen, but you get the same thing. So we have 64 possible ways we can put 3 nucleotides. So we have 4 nucleotides. Putting them together in a triplet, you get 4 to the power of 3. You get 64 possibilities. Again, the 64, what are they called? Trigrams in the I Ching. Whatever you want to think of. But it's called redundancy. It is the universal code because this code of the RNA coding for amino acid, the codon from the RNA coding for an amino acid, is true for every species on this planet. It's pretty cool. All right. That's all I can think of that I usually need to do on the board. And I'm sure I'll see you again. Bye-bye. Bye. Bye. And I'll give you some extra fairy dust magic. That you all are safe and healthy. Many blessings.
All right. Had to make sure you got that fairy dust blessings. So we're going to do practice. So I realized the problem I had with that is you couldn't see the code there. So for practice, you guys, I'm going to give you the DNA template. And so there it is with my ends labeled. And what I want you to do is make the complementary DNA, make the messenger RNA sequence, and make the peptide sequence. So if you've never done this, stop me, do it, and then I'm going to go through it. And so did you stop me? All right. So isn't that great? So that's just what was up there. I moved it down here. The complement, what was 3', we say 5'. So C complements G. Says, oh, you look amazing, G, with that extra tail there. A complements T. This is DNA. So A to T, T to A. Complementing. Oh, my. You look so beautiful today in your radiant purple. All right. Excuse me. RNA. This would be the RNA sequence. The info strands. So in pink, hot pink here, this is the key. This is, I put as hot pink. So the RNA always complements the template. That is how it is made. It is made off the template. So it comes out the same as the info strand. So the info and the RNA come out the same because they both are complementing the template strand. And the only difference is instead of a T, it has a U. So that's it. This is like the greatest code of all time. Now you can code anything, not just DNA. I do this with the kids when I teach the kids. All right. And then the peptide. So this is nice, much better than my video. So we look, the universal code is from the RNA. That's why there's a U. So always reading the RNA 5', that's why this says 5', we find GUA. So here's G. So we're down here in the bottom part, then U, and then it doesn't really matter. Anything you have there would be valine. So the third position in most of these codon triplets is often the redundant one. So again, this is G, here's U in this column, and we stay down here and then to A, valine. All right, the next one is CUC. So we're up here in this part. C we start with, we come here to U, and oh look, they're all Lu. So skip to my Lu. Right, and then AUC. So we're here, A, we come to U, and C is right there. There's my C is the second one down, and it says it's isoleucine. And then UGA. So U's up here, we go over to G, and we come down to A, which is right there. And it's stop. Yeah, and it's every place I've ever seen it, it's put in this order of UCAG because that was the order Nirenberg did it, and nobody's ever changed it as a respect to him. So a silent mutation, easiest would be with the valine if we just change GUA to GU anything. But I will say go back to the original template. So this position here is what became the A. If this T is any other letter, it is still going to be valine. That is a silent mutation because you don't change the amino acid. A devastating mutation is usually if you change the first position in the triplet in the codon. So if instead of this being GUA, it was AUA, we would be up here. So AUA, it would be isoleucine, which would change the amino acid. Now there's a little bit more to it because that wouldn't be a huge change because my knowledge, valine and isoleucine are both non-polar, so they'd both be in a hydrophobic pocket. It's more when you make a big change. All right. And that's actually going back one. That's one of the things that's was very devastating with sickle cell anemia. So I think it's an isoleucine becomes an arginine. So arginine is basic and it likes to be on the outside, and actually it's leucine. So the leucine, it's the second position. So it was Cu something becomes Cg something. And so it's a leucine got changed. One nucleotide was changed, and it changed the one amino acid from hydrophobic one, which was leucine, into a hydrophilic one, and the whole thing, the DNA, I'm sorry, the hemoglobin is then made where it doesn't fold correctly because the arginine is no longer scared. And it gives them a selection that the people who have sickle cell anemia, then of course they their blood doesn't carry oxygen correctly. The people who have no gene for sickle cell anemia, they actually get malaria. So that would be you and me. But people who have one positive and one negative gene, so they're heterozygous, so they have they don't express the sickle cell anemia, but they also don't get malaria, which is pretty cool. So it was a selection because malaria is a huge issue in Africa. All right, the anticodon is just 3 nucleotides down here. So the transfer RNA actually looks more like this beautiful purple and green figure. And down here, this is going to complement. It doesn't attach to the messenger RNA. It should say it is attracted. And again, that attraction is huge. This is all happening in under microseconds, in less than a millionth of a second. So the tRNA is attracted. This is the ribosome, this big green ball. Berkeley, University of Berkeley in 1960, when, yes, LSD was like given away in the cafeteria there. Anyway, they made this great video I usually show. And so you can Google it, translation Berkeley video, and you will get it. And so they did a play. It's about 15 minutes. And so some people are the growing amino acids, and some people are the ribosome, and they're rolling around on the field all together as this ribosome all dressed in brown. And then these people, there's a guy in a magician thing like my wand, and he gives them a poof of fairy dust, and the chemical bond forms. And it's actually a really awesome. It's one of the things I remember from when I took biochem. But the complement comes in, and these 2 tRNAs, and magic happens. A chemical bond forms, and then the next tRNA comes in, and then the ribosome keeps moving by, they call it translocation. It keeps moving by 3 nucleotides, excuse me. So again, there's the initiation where it comes together. You don't need to know all this detail. Your book gives you lots of detail if you want to read, and you can go online and learn even more detail, and it moves along, and this has to be covalent bond forms between the amino acids, and then you hit a point where we had to stop. Remember those 3 stop codons? The other thing that's really cool is they always start at AUG, always. Every amino acid sequence starts with methionine, AUG. Now, there is changes to amino acid sequence where we cut off some, and so the methionine gets cut off. There's extra sequence at the beginning, so the ribosome has to find this extra to find the beginning, and there's extra sequence at the end. So remember the telomeres, you can't ever get the very beginning and end, so there's always extra sequence. The RNA I worked on had a really long 5' end before the AUG, and there were 4 extra AUGs, and so that's part of what my research was 30 years ago, was looking at how there was regulation that determined if the ornithine was decarboxylated or not. Okay, so it had to do with extra stuff down here. So you can spend, years just looking at one RNA sequence and figuring out this one little piece of information. And so they say a PhD is where you learn more and more about less and less until you know absolutely everything there is to know about nothing. And you know what? That's the exact same thing that meditation is. So when you go deep inside, That's when you realize you know nothing. All right, recombinant DNA. This is really cool. So you can cut, we have enzymes that will cut DNA at specific nucleotides, and they make these sticky ends. And then you can cut, this is a bacteria plasmid, and you cut it with the same restriction enzyme, and then we can put any gene into there. And because bacteria always code, always transcribe and translate their whole DNA, it will now make this gene that came from somebody else. So the first one they did this with was insulin, and they were able to make human insulin inside a bacteria. And so the bacteria reproduces so fast, it doubles whatever every 15 to 20 minutes. And so we get exponential growth of it. And yeah, and then we go even bigger and we get into food that pretty much all your food's been, a good percentage has been altered. So it has a longer shelf life. They don't label it because the very first ones they labeled, and people didn't want to buy them because they didn't like the idea that their food was being tampered with by science. And that was before this whole craziness where everybody just eats junk food and gives junk food to their animals. And yeah, anyway, so you can go off on that train of thought in all your other classes. I'm going to go off on one last bit of information. So this is from The Private Life of the Brain. She said the reductionist genetic train of thought fuels the currently highly fashionable concept of a gene for this, a gene for that. So again, a gene codes for a specific protein, and oh my gosh, you can get your DNA tested and you can be like, oh my gosh, I have the gene for this. Oh my gosh, I'm going to get that. And if you believe that, you will. So the Central Dogma is a creed. That word dogma, it was a wonderful choice of word. It is a creed, and it is genetic determinism is where it went. I do not think that is what these gentlemen had in mind, that we would decide that our DNA code determines who we are, because it does not. Because the flow is not one direction. Yes, this is in a different language, because you cannot find this in the English language yet. We're that far behind. So actually, this is the new Central Dogma, or whatever you want to call it. I don't think they're that arrogant. So instead of saying it does go two directions, all these arrows go, oh, look at this one, the arrow goes both directions. Oh, and look at that, the protein is determining what DNA gets transcribed. So instead of having a one-way flow, it is actually a cyclic flow. Something to ponder. Are your genes really your destiny? That's Dr. Bruce Lipton, one of my heroes. And this is a quote from his book, which I'll show the title in a moment. Around the DNA is a protein sleeve. So these are regulatory proteins. The DNA is dormant potentiality, just like every atom in your body. It is potentiality until a signal from the environment starts the chain of events. Transcription, splicing, translation. What is this signal? That's the question. Then the gene moves from potential, just sitting there, it's a library book, just sat there on the shelf, until it's expressed, becomes the finished product, the protein. This is, he has 3 books that are out. He, again, is considered he's every year voted as one of the 100 most influential people, not in those magazines that talk about finances, which whatever money means, in the books about spirituality and stuff. Anyway, this is his book, Biology Belief. It came out in the 80s, maybe it was in the 90s. I can't recommend this book enough. It will completely change your perspective, especially if you think you know everything there is to know about DNA. Again, I have a doctorate in this, so I can pile it on higher and deeper than all of you. And then I read this book and I went, why don't they teach this? So you need to read that book. Which one is fact and which one's the myth? And then I went to Goodwill one day because they're the best book buying place you can, because you can buy books for $2. So this was the original cover of the book, the genie, right? So instead of just saying gene, the genie in our genes. So from our external environment, which is your diet, which we talked about, our friends, our support system, to our internal environment, every thought we think, every feeling and intention, our beliefs, our faith, our view of God. Some really cool studies they've done. People who view God as this judgmental, you've sinned, have a very different prognosis in how they recover from illness from people who have a view of God is love and I am love, and how they progress through diseases and stuff. All right. And really cool studies that have been done with all this stuff. All right, memories we associate with each are the tools of our consciousness. Each releases a cascade of biochemicals. You determine which cascade of biochemicals, and our body responds. Genes only contribute to the characteristics. They do not determine them. If you want them to determine them, that becomes your belief system and your consciousness, and then it becomes a determining factor. So you can also decide that you don't want them to determine. You can create who you are. You can be the genie in your genes. So yes, this is Joey. We went to the Oz Museum a couple years ago when we drove cross country. So this is from The Genie in Our Genes. This is a quote. He says, our body is a system of cooperation. It doesn't make a judgment about our feelings. It just responds. Our body cannot tell the difference between a real and a perceived imagined threat. And so I thought the Scarecrow in The Wizard of Oz is a good example of that, because, right, when they go through the whole thing of Oz and he thinks he doesn't have a brain, and it turns out he did the whole time. So it is your choice. Another really awesome book. This is quotes from this. The gene has become a super gene, a supernatural entity. It has the power to define identity, determine human affairs, dictate your relationships, and explain social problems. It's pretty amazing, because the media has given it this cult figure. Human beings in all our complexity are seen as a product of a molecular text. That's what we've allowed ourselves to become. Yeah, the secular equivalent of a soul. Or you can recognize you are the conductor of your genetic symphony. So yeah, this is, for those of you who watch my videos, that's the little icon up there. I had to clip out all the kids up here who were shooting marshmallows at me, which is what I was supposed to be doing in 2020 also on that day. And I will be again. And it was awesome. All right. So this guy also, their stories are amazing, because they talk about how they were really frowning and everything, and then they change their perspective, and they just are always smiling. All their interviews, great laughs. You all want to be like that. You don't want to be a person who's like grumpy all the time. You want to be that person who's happy and smiling, and you determine that. And so this is not mind over matter, it's mind to matter. So there's been so much emphasis, thank you, Sir Isaac Newton, that we are material beings, but your thoughts determine the material in your body. So that was Dawson Church. All right, highly recommend his book. So The Genie in Our Genes was the book I had the good fortune to read. So you direct your genetic fate. So you can keep it as, if you don't want to go where I just went, where I said your thoughts determine your genetic fate, you can just keep it with your food. You determine how long your telomeres are, the ends of your DNA. It's going to be a lab. You can change what genes are actually made by the food you eat. You can determine how many antioxidants are in there, which enhances, you can enhance DNA repair. And again, epigenetics is that you have the power over your health and well-being. You are in control of your cells and your genes. Your thoughts and environment heal you from the inside out. And that's just a reminder. That's me doing my kids' class as Tinkerbell. And I don't know what I'm saying to Tabitha there, but yeah, being of service, having fun, being playful, that also makes all the difference here. And so you determine that. And so, yeah, optimizing the health of your genes or genies. So we're not just beings of matter, we're energetic beings. We are actually energetic beings. And so I didn't get a picture of me dancing back there because I would have fallen on my face. And so that's me. We're sending rockets up at the college. I don't know if any of you guys remember what the college looks like, but it still looks like that. So every cell in your body, every cell in my body vibrates with energy and health. So loving yourself, loving myself heals my life, nourishing the mind, body, and soul so you can heal quickly. This is more real than that material world that we've created. So let your genes dance with your awareness. And this is a quote actually from the Buddha. So over 2,000 years ago, we are formed, molded by our thoughts. Those whose minds are shaped by selfless thoughts give joy when they speak or act. Joy follows them like a shadow that never leaves them. Yeah. And these are the people who always help me with the kids class. Because in all honesty, that's all I really care about. That if I get to go back on campus and be with the kids again, and you guys are all my kids too. So be playful. How can you be of service now? And anytime you're of service to others, you're also of service to yourself because your telomeres become longer, your DNA, your epigenetics shift. And so it could be just dressing up as Tinkerbell and going and standing, video chatting somebody who's down, and do it as Tinkerbell and send them magic. Or do it as, this is Joey here, as the one-eyed purple, he had a tail too, you can't see it there. All right, and there's Airy Berry Fairy, a very, very fairy fame. All right, many blessings. I have to figure out how to end this, and I'll see you at the next